So routinely will be happening. In lamb, there is a mutation of so-called myostatin, causing a special sheet called calipyg. What this means? Beautiful buttock, beautiful butt. You look at that, huge butt there. At the beginning, people think it's very good. There's a double muscle. Is that right? People think it's very good, double muscle. However, when you taste it, not good at all. Why? There is a low level of red fiber, muscle fiber. High level of white fiber and low level of fat. So that double muscle is not very good. You diluted it. Not good at all. Myostatin, we like it. If the cattle have that one, that's really very good potatoes. This one is not good. First of all, look at calipyg and normal. The calplastic. That's a calpain inhibitor. The amount is huge, almost double compared to normal. Second, look at the shear force value. Even you do aging, dry aging or wet aging, what happened? The normal one, shear force value from 21 days, 3 to 4. That's what we like, very tender. Look at the calipyg. 11 at the end of the day. 10. You can even, 8 to 9. I would say you're using a blade to cut, maybe not like a stone kind of. You don't even can do it. So that's example. So that means when you do the breeding for some of the sheep, these have to be avoided. Okay? We go here, talk about electron stimulation. Why we do electron stimulation right here? Now make sure this is different from electron stunning. Electron stunning, when you the cattle go there, they go there and then they like coma or like stun. This is when the carcass is already being finished. Then do the electron stimulation to make sure the pH decline and the rigor mortis happens more quick and to prevent the cold shock. Now what they really do? Speed up pH decline, cause adjacent tissue tearing, and the resulting contraction bands. There is two different levels of the voltage for the electron stimulation. Higher voltage improve the tenderness 20 to 25%. This is about 10%. So the voltage, you can see the difference. 550 voltage, low voltage only 35 to 50. So that's a function for the electron stimulation. Okay, if you don't want to do anything else, when the meter comes out, what you can do is you marination or injections of plant enzyme. Okay, there are many plant enzymes. Lots of them will do something for those enzymes. For example, papaya generate papain, pineapple have bromelain, fig will have fissin. They will be containing collagenous and elastins, will be break down collagen and elastin. Some of them could be injected into the meat muscle or marinated as a solution. All of them could be more effective if you are cooking at 75 to 85 degrees Celsius, which reach the internal temperature of the muscle. Something else we do. We can do adding some other chemicals. Sodium chloride, for example, phosphates. including improving water holding capacity, raising pH of the muscle. We can add calcium there, because calcium will be activated, calpain enzymes. And we also increase the calcium. We'll have impact for the structure proteins. And we also could be adding acetic acid, sodium chloride, can weaken the sarcolemma surrounding the muscle fiber. And I want to tell you one thing. Some of them maybe is the antimicrobials, let's say salt or acetic acids. People find that later on. At the beginning, people using them do try to make the meat is more tender. That's a purpose. Later on, they find they can kill bacteria. Okay, that's much later. So, injection with organic acid, salts, phosphate, calcium chloride will make the meat very tender. How about another easy way to make it? Just use bundle or blades or like needles to disrupt the muscle, myofibril, muscle fiber, collagen, all the proteins. A large portion, 50% of the United States food service beef is doing blade and needle tenderization. They are passing through 1 to 2 times, or will cause 2 to 8 times more tender. Dramatically decrease the share force value. This is easy to understand. Blade and needle, just cut. We can buy a normal, which is a portable one, only $50. I'll do that in my food micro lab. We can tenderize the meat. Okay? That's very easy. Break down the muscle. However, this comes out later. A major microbial safety concern during tenderization. If this needle is contaminated with E. coli 01578H7, it will be penetrated inside of the meat. If this solution, marinated solution, has been contaminated with E. coli 01578H7, this will come the problem. Because all of these beef, we call it non-intact beef, which means all the beef has been going through mechanical or chemical tenderization. E. coli 01578H7 will generate Shiga toxin, is an adulterant certified, regulated by USDA FSIS, which means ground beef, any of the non-intact beef, including beef steaks, you're not allowed to have 1 cell of E. coli 01578H7, and 10 cells will let people die. There is a lot of outbreaks happens back in 2000 to 2007. Lose $5 or $6 million. Most of them is inadequate cooking to law or rare status because we want tenderness. Some of them you cook directly from on-salt status. People miss the conception between the internal temperature and the temperature of the surface. Okay, so when I was a student back in Colorado State University, about like 15 years ago, we did a bunch of study about microbial safety of the tenderizing meat during the meat tenderization process. Because we always care about tenderness. At the end of the day, we find out microbial safety concern is a major issue. So roughly cooking to the internal temperature, 71.1 degrees Celsius, is regulated by USDA to make sure the food safety of the meat. Okay? So that's all I have today for the tenderization. So I think these part, which is talking about a little bit of meat science, is important. Okay, then you can understand this material very well. Okay? Thank you.